What if aging wasn't a slow and uniform decline, but a mosaic of cellular rebellions? Some cells giving up, some hanging on, and some that could be brought back from the brink with a drug. That's not a metaphor. That's the emerging science of cellular senescence. And today we're diving into a study that cracks open the black box like never before. Welcome back to Decoding Longevity, the podcast where we explore the science shaping the future of health and lifestyle. I'm your host, Raghav Sehgal, with my co-host, Macsue Jacques.
Hey everyone! In our last episode, we have unpacked in detail the roadmap for translation of aging biomarkers from bench to bedside. But today, we are looking inside the aging cell itself and exploring in more detail one class of aging drugs that is highly studied. And for that, we have a guest that will help us understand what goes behind the scenes and explain how these drugs actually work.
Dr. Iman Azimi, a scientist who's pushing the boundary of how we understand and potentially treat cellular senescence. His recent paper titled Single-Cell Fluorescence Imaging Reveals Heterogeneity in Senescence Biomarkers and Identifies Rapamycin's Responsive Subpopulations is a game changer. Iman, welcome to the show.
Thanks a lot. Thanks for having me. Pleasure to be here.
That's great. Well, let's start a little bit with the basics, but it's exciting nonetheless. So most people in longevity circles have heard of senescence. So the idea that cells can stop dividing but not necessarily die. Instead, they secrete inflammatory signals that drive aging. But your work flips the script a little bit. What did you find when you actually looked closer at these so-called senescent cells? Can you maybe give a little bit more insights into those zombie cells?
Absolutely. While the idea of zombie cells paints senescent cells as a uniform, misbehaving group, what we found is far more nuanced. We used high content single cell fluorescent imaging and discovered that even within a population of cells exposed to the same stressor, like chemotherapy, for example, senescent isn't one fixed state. Some cells crank up inflammatory cytokines like IL-6, others swell in size, and some show high level of P21 expression, and a few even continue low level DNA synthesis. These cells aren't just stalled, they are diverse, dynamic, and likely play different roles in aging and disease. So rather than a single zombie state, senescent appears more like a spectrum of phenotypes that evolve over time.
Yeah, that's really interesting. And so I particularly love the fact that you use sort of image-based single cell profiling in this case. And it feels like a bridge between sort of old school microscopy and high throughput omics. But why did you really choose this particular path to go down? There are so many other approaches. Why did you choose this particular route?
Yeah, that's exactly the bridge we wanted to build. Traditional methods like Western blotting and qPCR provide only population level averages. On the other hand, methods like flow cytometry, although a single cell is often limited in a spatial context and can struggle with complex morphological features or overlapping signals. By combining high content microscopy with single cell analysis, we could capture not just marker expression, but also the cell shape, size, and the spatial relationships. And this is all in one snapshot. The approach allowed us to uncover subtle subpopulations that would otherwise be missed. It's quantitative, it's scalable, and importantly, embraces the complexity of senescence.
Yeah, so that's very interesting. So I assume in this technique itself, you end up getting, a lot of cells. Did you choose a specific tissue to look at, or have you looked at many different? What kind of cells did you actually, and how did you get to that senescent state to be able to image them? How did you did the selection?
We initially used, and the cell type that's mainly used in this project in the paper it's in bioarchive now. It's primary human dermal fibroblast, and these are really commonly used cells in senescent and aging studies. And the model we used to induce senescent, there are multiple models that we used. One is natural senescent models, that is also known as replicative senescence. Senescence will cells replicate until they lose their ability to replicate further. This is called Hayflick limit. That's been known for many years. So this is a kind of natural replicative model. We also use accelerated model of senescence, which is by using chemotherapy. For example, we use mitomycin, known chemotherapeutics. They causes senescent via DNA damage. We also used metabolic way of inducing senescent, which is using D-galactose, which causes oxidative stress and causes senescent. So there are multiple models. And we also used other types of cells rather than fibroblasts, which is human epithelial kidney cells or arptic cells. And those are non-fibroblast cell types. So it's kind of diverse method and diverse cell types.
Yeah, I think one of the really interesting things in the paper was the fact that you suggest that at this point, not all senescent cells are equal. In some ways, most clinical trials kind of assume that's with Senolytics assume that all senescent cells are equal. Your paper suggests that's fundamentally flawed. Can you talk about that a little more?
Yeah, absolutely. That assumption that all senescent cells are equal and can be eliminated with a one-size-fits-all approach doesn't really hold up in our data. And we saw a striking heterogeneity in biomarker expression, even within a single senescent population that persisted across different stressors and cell types, even in synchronized cells. So it wasn't necessarily just cell cycle differences. And for instance, we tried rapamycin, it's a known xenomorphic, and we found that it's not a blunt instrument really, and it's quite selective. It didn't suppress all senescent markers equally. For example, it was effective at reducing nuclear area and P21 expression in some subpopulations, but had minimal impact on others. And similarly, it didn't target our senescent populations and rather specific subpopulations. Interestingly, its effect wasn't confined to senescent cells. It also altered these markers in proliferating cells. Rapamycin as xenomorphic potential, we found that it doesn't uniformly tame senescence. Instead, it reshapes parts of the phenotype and similarly impact some cells more than others. And this is the same for Senolytics, also impact those Senolytic studies. And knowing that it's not just one senescent population. When we continue designing Senolytic trials, we cannot assume all senescent cells are equal. And if we do that, we risk missing the mark. And probably just like in cancer, that is stratified based on molecular subtypes, we may need to stratify by senescent subtype or molecular profile to really tailor treatment.
Yeah, I guess this is really important, for biomarkers as well, and potentially the reason why some senescence treatments haven't worked as well as we hoped they would, because it wasn't considering all this diversity. And potentially, I guess, the pathway forward, as you mentioned, you use rapamycin to treat some of those cells, but not all of them responded the same. So would you agree, or what are your thoughts in relation to, say, using a combination of drugs? Have you been able to come across anything that worked a little bit sort of like better than the rapamycin, or, that could target different cells at the same time, or...
Yeah, that's a really interesting concept, and we may need that sort of combination approach to tackle different aspects of senescent. For instance, could be the aspect of replication of senescent could be quite different to the aspect of inflammatory side of it. And we have actually identified some drugs that they specifically control that inflammatory side of senescence, which is really known to be a bad side of senescence, really, that inflammatory drives a lot of pathogenesis and disease progression and initiation. And we may actually have some good and bad senescent, and not all senescent cells could be actually bad, and some can have beneficial roles. So combinatory approach is really interesting, and we have also identified some compounds that really specifically drive, I mentioned the inflammatory part, and we have some compounds that drive just the, for instance, cell cycle protein expression like P21 or P16. So a combinatory approach could be really interesting and see to sort of tackle this heterogeneity of senescent and also different markers.
Just before we go to the next question, let me just kind of hook in here. You mentioned good and bad senescent cells. Do you mind talking a little bit more about like what would be a good senescent cell here? That's very interesting.
Yeah, absolutely. So this is still something that we are working on to really identify what are these senescent subpopulations and what they mean. But we have identified some senescent cells that they don't necessarily have that typical inflammatory profile. And that could be something that can define them as not really harmful senescent cells. But this is something that we are going to address in the next research question to really identify if a cell is senescent, at least have the typical cell cycle arrest and replication stoppage, but it doesn't have that inflammatory phenotype. What does it really do, and what's the impact on the neighboring cell? Can we really consider it as a harmful senescent or not? That's just a hypothesis at the moment that we have, and we are going to test.
I think you also pointed out something really interesting in this conversation around the good versus bad, but also sort of instead of targeting the senescent cells, which is what Senolytics do, having sort of the inflammatory markers being targeted, for example, which is what now is being categorized as xenomorphics, altogether a different category of therapeutics. How do you see sort of this debate between xenomorphics and Senolytics evolving, and how do you see sort of your technology aiding and abetting in that debate?
These are called, yeah, as you mentioned, under the umbrella of Therapeutics. We are mainly divided into xenomorphics or senolytics. The idea of senolytics is to eliminate these senescent cells selectively without harming normal replicating cells, healthy cells. And the idea of xenomorphic is just to sort of suppress that senescent or delay or reduce that inflammatory profile of these senescent cells. And that's something that there are two different approaches, very quite differently. One you want to eliminate it altogether, and one you want to suppress its sort of inflammatory profile. And this is something that we are interested in both these approaches, and they can be used in different contexts. And we are actually testing compounds at both xenomorphics and other compounds that there's senolytic sort of capability. And this can be different we don't know what it's really, we have to test it in different disease contexts. Senolytics are generally known to have more side effects because you're killing cells, right? And that could have some side effects and clinical or even preclinical models have shown that they do have. But again, choosing the right senolytic that is really selective is really important. And it needs to be tested in different disease phases. The skin aging could be different than in fibrosis, for example, and one approach could be better than the other in certain diseases.
Have you been trying different diseases? Sort of like, where do we go from here now? I know that this is, you just started using this technology now, having the live cells and being able to identify those different cells. But what would be, there's new research questions, but thinking about going from the lab or the bench side to the clinic, what are the next steps and where do we go from here?
There are multiple things. One is that sort of disease model that you mentioned, and at the moment we are working on multiple pathological models, which is skin aging. We're working on that. We are working on wound healing, and that can be actually a good side of senescent because in wound healing you have acute senescent and chronic senescent. Acute senescent is actually good for wound healing. That sort of secretory inflammatory helps the wound to heal, but chronic senescent, that existing old cells, is actually not good for wound healing. So we have that model. We have model of fibrosis, which is cardiac fibrosis and also kidney fibrosis. We have these two models too. And we also have another aspect of senescent, which is in cancer and therapies that used in cancer, like chemotherapy, radiation, and all that. They're actually supposed to kill cancer cells, and they do kill cancer cells, but not all cancer cells. And they induce senescent in a population of cancer cells. And these cells are the ones that can secrete that inflammatory, they can make the surrounding cells more aggressive, they're resistant to therapy, and they can actually cause tumor relapse. So we're also working on the cancer model and specifically in brain cancer. So the next step in terms of what we have found at the moment, apart from these disease models, is to really understand the nature of these heterogeneous senescent populations. Why are they different and what drives that diversity? We suspect it may be linked to the inflammatory profile. And as I mentioned, it's possible that not all senescent cells are harmful and some might actually play beneficial roles while others drive disease. So distinguishing this sort of population is critical, and if you like, good and bad senescent, although at this stage it's just hypothesis. We're also interested in why cells exposed to the same stressor respond so differently. Some recover, some die, and others enter varying degrees of senescent. What determines that fate? We think tools like machine learning could help us uncover patterns that predict how cells respond based on the molecular signatures. At the same time, we're also working on developing novel single-cell based assays that can be used in high-throughput drug screening platforms. And the goal is to discover new xenotherapeutics, either xenomorphic or senolytics.
That is really fascinating. Yeah, it feels really like that we are in the verging of making senescence not just something we fear, but like something that we can manage and we really beginning to understand. So with more precision. So, and maybe as you said, potentially reverse it. I know that there's some experiments maybe going in the lab looking at reversing the senescence. Iman, thank you so much for joining us and for sharing a little bit of your work. I know it's a lot, and this is just a tiny snapshot. But I know that this paper has really shifted, the understanding. And thank you so much for your contribution. So I'm very excited to see where this is going to lead.
Thank you so much for having me. Really enjoyed it. Thank you.
Thank you all for listening. If today's episode sparked your curiosity, be sure to check out Iman's paper on Bioarchive. We'll link it in the show notes. Until next time, keep decoding longevity. Thank you.
Thank you. See you soon.